Now, up until now, I've been keeping things very, very simple. I've said if these traits are on the same or different chromosomes, they might be passed on together or independently. But in fact, genetics is quite a bit more complicated than that. And this is because of a phenomenon called crossing over. It's all part of recombination, where we get a whole bunch of new combinations in our gametes. Now, here we have our sister chromatids for homologous partners. So this could be chromosome number 1. Okay, let's say this is chromosome number 1 that came from mom. And here's chromosome number 1 that came from dad. So they've each undergone DNA replication, so they're sister chromatids. The sister chromatids line up together to form the tetrad, because this is going to be the beginning of meiosis. Okay? And what I want you to imagine is that in your body, you have had one chromosome number 1 from your mom and one chromosome number 1 from your dad. They've been living in each one of your cells perfectly well, okay? And in your body now, you're going to undergo meiosis. A few cells are going to undergo meiosis to produce your gametes, okay? And it's like a last dance that mom and dad's chromosome number 1 have in the cell that's about to undergo meiosis, okay? And they're going to hold hands one last time in there, okay? But when it comes time to separate, there's actually been a recombination, a crossover. So mom's long arm here, she's now got dad's hand from the end of that chromosome there, and now dad's long arm has got mom's hand there. So these are now going to be in new combinations that ever occurred before because of these crossovers, okay? So this is yet another way we get phenomenal genetic variation from one generation to the next through meiosis. You already had the independent assortment that these whole chunks of chromosomes are not being passed on in tandem. We saw that you had a phenomenal amount of variability in the gametes produced by mom and by dad. You put that together in random fertilization, like 1 in 70 trillion. Okay, now we're going to have crossovers. So we have the tetrad forming, okay? The sister chromatids are having this one last dance, mom and dad. They end up with different bits and pieces separated off. And so now you've got, whoa, wait a minute, that wasn't even there before. You've got mostly blue, well, you've got all blue chromosome on this one here, this sister chromatid, but this other one's got bits and pieces from the other parent, okay? And there's the reciprocal here, and then there's yet another one that's the whole chromosome from way back here. So there's all these different combinations just in one meiotic event at this one pair of chromosomes, okay? And if we actually look with an electron microscope at what happens with the tetrad, there really is this dance. The two sister chromatids get quite close together, and there's crossovers that occur not just at the end, but all through the chromosome. And these are called chiasmata. It's not a term you really have to know, but it's just a physical place on the chromosome where these crossover sites are most likely to happen. Okay, let's look at this again, another diagram. What we're going to have is long chromosomes here, a sister tetrad for a long chromosome, and here's a sister tetrad for a short chromosome. So the homologous partners have found each other with their sister chromatids. We've got a tetrad here, got a tall tetrad up here, okay? And we got this thing going with the dance. The crossover is happening, these chiasmata, there it is down here, here, and here. As we go through these phases, we're going to see that those are being preserved. Okay, those crossovers, that's a really important thing that goes on, and it continues to keep them intact. So we have mostly red with a bit of blue, mostly blue with a bit of red for both the long and the short sister chromatids after they split apart from the tetrads. Going out, we just care about the bookkeeping. All that matters to us is that as we go through and we finish meiosis, there we go. There's going to be separate, we're going to be back to the haploid state. And the gamete here, there's a sister chromatid separate. We've got in those daughter cells, we've got a recombinant chromosome here. And here's another one that didn't happen to have any recombination there, but in another sperm cell, and another egg cell here, there is recombination in that long one, not in the short one. So each gamete, in just about every chromosome, there's a crossover as well, okay? So each chromosome and each gamete has at least one crossover. So if we have loosely linked genes, or loci, okay, on the same chromosome, so we have this trait here, alternative alleles are red and blue. This trait here, alternative alleles are green and brown. In this chromosome, the red allele is with the green allele, and on this chromosome, blue was with brown, okay? So these are further apart on the same chromosome, and it can happen that they might get split up by a crossover, okay? So that in their gametes, now we've got something that didn't occur in the parent. We've got a single chromosome that now has the red with the brown. That didn't occur over here. The blue with the green, that didn't occur over there, okay? So we've got enough distance that these crossovers may actually separate these different alleles at the different traits, okay? That's recombination. Now, if we have non-linked genes, let's do a really, really simple breeding experiment here, okay? We're going to have 4 equally likely combinations in gametes of a dihybrid, okay? We're going to have the 4 different alleles. So these are 2 different traits. We've got the long chromosomes code for trait A. The short chromosomes code for trait B. On this chromosome is allele capital A, the dominant allele. On this long chromosome is the recessive, likewise dominant and recessive. Okay? So these are heterozygotes. Okay? So we're going to look at the gametes of this individual, and they should be equally likely because of independent assortment. They're on separate chromosomes. Okay? We should see capital A with capital B just as often as capital A is with little b, and little a is just as likely to be with capital B as it is with little b. Okay, so those are the 4 combinations that we saw with independent assortment when they're on separate chromosomes. Okay? Now, this is just an accounting thing. What we want to do is to do a breeding experiment where we cross one of these with a double recessive. This only has recessive alleles, okay, for the A trait or the B trait, all recessive. Okay, what we should then see in the offspring is an equal proportion of these different types, okay? A double dominant will be 1/4 of them, a single dominant for the capital A, a single dominant for the capital B, and then a double recessive, okay? So these have the 4 different possibilities of gametes from this one. These are all always the same, okay? So capital A with little a is going to be heterozygous, capital B with little b is going to be heterozygous. So you get that genotype in the zygote, you get this genotype in the zygote of these, and so on. They're all equally likely, okay? This is just a simple accounting system. Now, let's do an actual cross, okay? Let's take 2 traits that we're interested in, and we know this is a double heterozygote, okay? It could be anything. It could be a widow's peak, and it could be, you know, the attached earlobes. It could be anything we wanted, okay? Any 2 traits. We're going to cross it with a double recessive, okay? And we expect these 4 different possibilities, okay, to occur. And what we observe is 45% are going to be double heterozygotes, and 45% are homozygote recessive, double recessives, okay? And 5% where there's just one dominant trait, recessive in the other, and the other 5% are the inverse of that, okay? Now, if these traits had been truly unlinked, as Mendel's peas were, okay, and we'd done this, as I'd shown in the previous slide, there should have been all four combinations just as likely, okay, and these would always provide you with that background of a recessive, so you could see the genotype from the phenotype, and we would have expected a ratio of 25%, 25%, 25%. They all would have been equally likely if, as in the prior example, there had been independent assortment, if they'd been on separate chromosomes. On the other hand, if that capital A and capital B allele were on the same chromosome and they're very close together, they wouldn't be separated by recombination, by crossover, so they would have been passed together in tandem. So if they were perfectly linked, half of them should have been double heterozygote, and the other half should have been double recessive. But that's not what we saw. This experiment here, we've got a few that we cannot account for. There's 5% that's dominant for only one trait, and 5% that's only dominant for the other trait. And we don't expect that if they're perfectly linked or if they're perfectly unlinked. Okay? Now, you must be thinking, God, this is tedious beyond words. And it is tedious, no doubt about it. But there's something amazing you can do with this information. You can start to map where traits occur on chromosomes. So here's the result of a lot of breeding experiments in Drosophila, a fruit fly. And there's alternative forms for lots of different phenotypic traits. So we have some Drosophila that have these aristids, like their antennae, that are long versus short. Some that have nice long wings, some that have very short wings. Some that have long legs, some that have short legs. Some that have red eyes versus purple. Long versus vestigial wings, red versus brown eyes, okay? Now, if we cross some of these, tracking two traits at a time, we begin to realize something, that if you have a fly that has both long antennae and long wings, okay, or you have the short antennae or dumpy wings, these traits do get passed on in tandem, okay? Not perfectly, but almost always passed on together, okay? Really tight linkage. Whereas if you take long versus dumpy wings and then do a dihybrid cross with that, tracking the color of the eyes as being red versus purple, these are not always passed in tandem, or even not often passed in tandem, but sometimes they seem to be passed in tandem, more often than you expect if they're on different chromosomes, okay? And you go through all these and you find that, whoa, these things are showing linkage, they must be close together. And as you go from one end of the chromosome to the other, if you did a dihybrid cross on long versus short antennae, and at the same time looking at red versus brown eyes, these would behave as if they're perfectly unlinked. But it's actually because they're opposite ends of the same chromosome, okay? Now again, this may seem tedious. This may seem like, oh, crumbs, can we move on to another topic, okay? And it is tedious, I agree, it's really tedious. But the fact is, if you do things like this, you begin to understand so much more about how our bodies are actually built. And so, nobody deliberately breeds people according to traits in question, but families, marriage happens pretty much at random in a lot of respects. The traits that we choose, our wife or our husband, are not all that similar to ourselves. And you can use a pedigree analysis in humans to determine the precise locations of specific genes. Now, in this day and age, we have the entire sequence of Gattaca for all of our chromosomes has been decoded. We know what that is, okay? But we don't really know what they do, okay, unless you do something like this. And if you track certain traits, inherited diseases, for example, like amyotrophic lateral sclerosis, which is also known as ALS or Lou Gehrig's disease, turns out that that's on chromosome 21. It's in this region of chromosome 21, and that's codes for a protein. If you translate and transcribe and translate the DNA over to the protein, that's the enzyme superoxide dismutase. So now we know, because of these kinds of pedigree analysis, whoa, those are people that have a problem on chromosome number 21. We can work out from that what might possibly be a suitable treatment. Here's another genetic disorder. We're going to see this in a few lectures. It's a very sad one that causes premature aging. Small children are old by the time they're only about 12 or 13 years old. And this turns out to be coded on chromosome 8, right down here. And so right now, people are trying to find the proteins that are coded down there that could be used as a genetic therapy. So with the Human Genome Project, by going through and getting the Gattaca all the way along, all these different chromosomes, for the 3 billion base pairs in our genome, we now know that that codes for about 25,000 different genes, 25,000 different proteins. And by putting together all that Gattaca information with the kind of linkage maps from these pedigrees that people have been doing to see how they do or they don't pass on, showing signs of linkage or not, it's been possible to place certain proteins along these genes associated with certain diseases. And so we've sometimes seen that this refers to 3 billion base pairs. That's the haploid genome. That's like going along, getting the Gattaca along a single sperm or a single egg. But a diploid genome, you have homologous partners. So we all have 6 billion base pairs coding for those 25,000 genes, but we have 2 alternative forms if we're heterozygotes, or 2 of the same if we're homozygotes for any given gene. Now, one of the people that led this is a particularly feisty molecular geneticist named Craig Ventnor, and it turns out that the first map that was ever developed of a diploid was of his own genes. And he did this because a lot of people are worried about, well, if the government knows my precise genes, they might have some impact on something about my life, or insurance companies might be less or more likely to give me insurance because I carry around certain alleles. He didn't worry about that. He's a wealthy man, so he didn't care about getting his insurance, and he's powerful enough he didn't care about any kind of government stuff. And so here are his genes, okay? And it's kind of an interesting story in that when he published this, people were very interested to see something about his genetic future. So he carries around certain alleles on the chromosome number 1, SF4 that's linked to heart attacks, which may make him more prone to heart attacks. He also carries on chromosome number 2 a lactose intolerance allele. So he's more of a night owl because he's got an allele here on chromosome number 4 that does that, etc., etc. But what a lot of people found quite amusing about this story is that he's a really obnoxious man, stepped on a lot of toes to push ahead with the human genome sequence himself. And it turns out that on his X chromosome, he has a particular allele called MAOA that really basically turns you into a son of a bitch, okay? So that's Craig Ventnor, and that's his genome.